Cell signaling in Creutzfeldt-Jakob disease. CJD is a rare degenerative disease in the brain. Although the symptoms are neurological, such as the loss of intellect, personality changes, and speech, and resemble those of dementia and Alzheimer's, Creutzfeldt-Jakob disease progresses significantly faster, with a 100% fatality rate. CJD comes in several forms. Sporadic is the most common, occurring in 85% of cases. Hereditary is based on familial history, occurring in 10 to 15% of cases. Acquired CJD can also occur when infection follows a medical transplant or ingestion of infected meat, which happens in less than 1% of cases. The regular signaling pathway involves the GPCR protein coupled receptor that uses calcium and IP3 as secondary messengers. The PRNP gene codes for the prion protein, PRP. The cellular prion protein, PRPC, can act as a ligand to activate the G protein coupled receptor and then the G protein, which replaces GDP for GTP. The G protein travels to activate the phospholipase C enzyme, which cleaves a plasma membrane phospholipid called PIP2 into diacylglycerol and inositryl triphosphate. The IP3 travels through the cytosol and binds to the IP3-gated calcium channel in the endoplasmic reticulum membrane. As a result, calcium ions from the lumen of the endoplasmic reticulum travel to the cytosol. Calcium ions are secondary messengers and activate various proteins, which in turn produce cellular responses. CJD causes a mutation to occur in the PRPC protein, misfolding it into a PRPSSC protein. PRPC is non-infectious and mostly ahelical in structure. PRPSC is infectious and consists mostly of B-pleated sheets. PRPC is synthesized and processed in the endoplasmic reticulum and the Golgi apparatus. It then attaches to lipid rafts in the phospholipid membrane. As more PRPC is produced and recycled, the misfolded PRPSC prion attaches to the membrane. Its origin depends on the type of CJD. It could come from a lysosome and an exosome, or from cell-to-cell contact. It causes more PRPC to attach and causes them to misfold as well. As the chains grow, the PRPSC breaks off and spreads. This process takes place inside the membrane of a nerve cell. If the PRPSC binds to the receptor, it would stay and cause the signal to be continuously activated. This would lead to a constant release of calcium ions into the cytosol and cause cellular stress. The nerve cell synapses would be impaired as calcium is important in neurotransmission. Cell apoptosis will follow. There is no cure or ways to slow the spread of Creutzfeldt-Jakob disease. Therefore, treatment focuses on supportive care and relieving pain in patients.